Good morning. My name is Janie Montblanc, and on behalf of the Great Basin Fire Science Exchange team and partners, I would like to welcome you to this webinar titled Seed Zones, Development and Use, Procurement and Deployment, and Provisional Zones for Native Plants, presented by Brad St. Clair and Vicky Erikson from the Forest Service Pacific Northwest Region and Andy Bauer from the Olympic National Forest. Before I introduce our speakers, I want to let you know that you may ask questions of the speakers or me at any time during the webinar by typing them into the questions pane of your control panel at the top right of your screen. I will keep questions for the presenters in the queue and field them after the presentation, although some clarification questions may be addressed after each presenter. I also want to let you know that whatever you do in your control panel does not affect the webinar presentation, so you are welcome to type a test message or test your audio at any time during the webinar. Now I would like to introduce our presenters. Brad St. Clair is a research geneticist with the U.S. Forest Service Pacific Northwest Region in Corvallis, Oregon. His research is primarily aimed at understanding the genetic basis of how plants are adapted to their environments, and he is developing web-based tools that will help managers choose appropriate seed sources given different climate change scenarios. Andy Bauer is the area geneticist for Western Washington and Northeast Oregon. His work involved genetic resource management on national forests, and he is also the project lead for the Forest Service Whitebark Pine Restoration Program for Oregon and Washington. Vicky Erikson is the Regional Geneticist and Native Plant Program Manager with the U.S. Forest Service Pacific Northwest Region in Pendleton, Oregon. Her work includes developing and managing gene conservation and operational seed production programs for conifer species and grass, shrub, and forb species used in restoration activities on national forests and grasslands in Oregon and Washington. Welcome, Brad, Andy, and Vicky, and thank you for being here today.
Well, thank you.
All right, I'm making you the presenter.
Okay. Okay, I think we're all set then. Okay, so welcome everybody to this first webinar in this series titled The Right Seed in the Right Place at the Right Time, Tools for Sustainable Restoration.
Sorry to interrupt you, but your slide is a little bit cut off.
Okay, should I relaunch it then or something?
You could try, or maybe just try, oh, there we go.
There we go.
Got it.
Okay. So the focus on this webinar series is on how to choose, acquire, and use appropriate plant material to meet the goals of sustainable restoration. And the topic of this first webinar is seed zones. So seed zones are a tool to ensure appropriate plant materials, that is the right seed in the right place. Today you'll hear from three of us. First I'll provide a little background on, there we go, three of us. So first I'll provide a little background on seed zones and then talk about how we come up with seed zones using information from genetic studies. We sometimes call these empirical seed zones. Second, Andy will talk about the development and use of seed zones for species for which we lack genetic information, and we've called these generalized provisional seed zones. And third, Vicky will talk about her experiences and those of others in the Forest Service in the Pacific Northwest on using seed zones to guide procurement and deployment for native plant material. But first I'd like to say a few words about the primary objective of this webinar series and much of all our work, and that's sustainable restoration. So the Society of Ecological Restoration defines sustainable restoration as an intentional activity that initiates or accelerates the recovery in the ecosystem with respect to its health, integrity, and sustainability. So when we talk about restoration and recovery, we're most often referring to returning an ecosystem to some semblance of an earlier composition, structure, and function, or as close to it as possible given changed circumstances like warmer climates. And when we talk about sustainable, we're talking about maintaining that health, integrity, and productivity of ecosystems in the long term, in perpetuity. So what we're not talking about is just simple revegetation, returning plants to the landscape. And we're not talking about rehabilitation, which is sometimes used synonymous with restoration, but sometimes refers to a more narrow range of objectives. And we're not talking about creating what's been called novel ecosystems. So novel ecosystems have more of a focus on some human-defined function with less interest in earlier composition and function, which is fine if that's what is desired by managers and decision makers, but I wouldn't call that restoration. So with restoration, the objective is to return an ecosystem to some appropriate historical norm or as close as possible, since we all recognize that environmental circumstances may have changed. So in this first part, I'm going to talk about the development of seed zones. I'm first going to give an introduction to the idea of seed zones, what they are, and why we want to use seed zones. Second, I'll give a brief history of seed zones and the development of early seed zones in the last century. This is mostly from a forestry perspective. That's where they were mostly used. And third, I'll talk about how we've come up with seed zones more so lately using genecology studies, and then also a little bit about reciprocal transplant studies. So that's the outline for this first part. So why seed zones? Well, the short answer is because we care about the near and long-term adaptation of plant populations. In the near term, we care that plant populations have good establishment, high vigor, and are capable of reproduction, that they're adapted. In the long term, we care that they continue to evolve through natural selection to be adapted when there's a change in the environment or a new stress or a new disease. This depends on genetic diversity, a function of genetic diversity, and seed zones help us to maintain that natural genetic structure and diversity. So a seed zone is an area within which plant materials can be transferred with little risk that they will be poorly adapted to their new location. So we have some sort of environmental gradient, and the assumption is, and then we have a genetic line, and the assumption is that natural selection has resulted in genotypes in a population that on average are adapted to or match the environment. So the assumption is that plant populations are locally adapted, and I'll say a little bit more about that later. And so what a seed zone does is simply draw a line somewhere along that gradient that says we can move populations, move plant material within that zone, and it should be reasonably adapted. It will survive, grow, and reproduce. But we really are not supposed to move things across that zone, even though they may be similar. So there's another concept related called seed movement guidelines or population movement guidelines, which are directions or rules for moving specific populations to new locations and still ensure that the plant material is adapted. We sometimes call these continuous seed zones, or another term that's been used is focal point seed zones. So that is, we can move, so in this case, the population guidelines do not necessarily draw a line, but instead provide a rule that says you can move them anywhere along that gradient, but only a certain environmental or corresponding geographic distance. So population movement guidelines are probably more appropriate given clinal variation, but they require a little bit more managing, less bulking of populations and keeping track of more seed lots in the collection, and increase in deployment of the plant material. So the other reason we have seed zones is because our policy tells us so. So for example, in the Forest Service manual, we're instructed to ensure that genetically appropriate native plant materials are given primary consideration, and then it goes on to define genetically appropriate as adapted to target site conditions with good establishment, vigor, and reproductive capabilities. Genetically diverse to allow adaptation to changing climates and environmental conditions. Unlikely to cause genetic contamination, not likely to become invasive, not likely to be a source of non-native pathogens, and is likely to maintain critical connections with pollinators. And we see this term genetically appropriate pop up all over the place. We also have a reforestation policy that says to not use seed and seedlings of exotic tree species or native species from an off-site source except where scientific studies have proven them to be adaptable, or administrative studies or tests are being carefully planned with cooperation or assistance from research scientists. And the Department of Interior has similar policies. In fact, just at the beginning of the year, there was a news release from the Interior Secretary Jewell on strategy to protect and restore sagebrush lands threatened by rangeland fire, largely driven by concerns about sage grouse. And in Section 5, it says, to the extent practicable, utilize locally adapted seeds and native plant materials appropriate to the locations, conditions, and management objectives for vegetation management and restoration activities, including strategic sources for acquiring, storing, utilizing genetically appropriate seeds and other plant materials native to the sagebrush steppe ecosystem. And seed zones are a tool to get there. And there's other policy directives from agencies or other organizations. And for example, the BLM Oregon Washington genetics plan says to maintain the integrity of natural genetic structure. Other organizations are a little more conservative. The National Park Service says it's closely related genetically and ecologically as possible, preferably from adjacent or local areas. And we see this in other organizations too. So what I'd like to do now is just talk a little bit about the history of seed zones. Now, this idea of geographic structure and adaptation and concerns about moving things is not new. In 1971, Langley published a review of 200 years of genecology dating back to the mid-18th century, talking about provenance trials and moving things around. So the idea is not new. In the Pacific Northwest, we started to become concerned with seed zones when we started to move tree populations around. Particularly, there was a large burn in 1902 called the Yakult burn. It burned 300,000 acres, and they began to be concerned about reforesting that burn, and so the Wind River Nursery was established in 1908, one of the first nurseries in the Forest Service, to help produce material for that. And at the same time, there was still interest in this question of using exotics or questions about seed source. And so two pioneering research studies were established at the Wind River, next to the Wind River Nursery. The Wind River Arboretum, which tested species from all over, and the Douglas Heredity Study, which is essentially a provenance test that looked at moving things around. But meanwhile, they began to see that some of the stands next to these naturally regenerated stands were not producing, were not as productive as the naturally regenerated stands. So we started to see early observations of poor growth and survival, and started to begin to get concerned about the idea of moving populations around. At the same time, the Wind River Arboretum showed that the exotic species generally perform poor relative to natives. Another observation that they saw in the Doug Heredity Study and the Wind River Arboretum is that maladaptation often took some time to develop and often resulted after extreme climatic events. So this led to the first seed movement guidelines in 1939, and those guidelines said that the Forest Service was going to restrict movement to 100 miles north or south and within 1,000 foot in elevation, with consideration for unusual climates, topography, or soils. Whoop, I went to the beginning. Just a second. So then in about 1960s, a group of geneticists and others got together and said, well, I think we need to outline these seed zones. So they developed a certification system, more detailed maps and guidelines for Oregon and Washington, and for California in 1970. This was based primarily on the collective knowledge of climate and vegetation types. And basically they sat down and said, well, to the best of our knowledge, these areas are similar, and drew lines around them. But they also included about 1,000-foot elevation bands within these geographic zones. So restrictions remained with respect to elevation. And then these were followed by various Forest Service guidelines published in the 1970s and 1980s for other places in the West and in the South. Meanwhile, we began to get knowledge on the genetic structure of different tree species, largely from the Genecology studies by Bob Campbell, Frank Sorensen, and Jerry Rayfeld. And based on this knowledge, the seed zones were revised in Oregon in 1996 and in Washington in 2002. And in the revisions, they basically differed by species because they recognized that some species, like Douglas fir, were more tightly adapted, were specialists, whereas other species like western red cedar were generalists. And so we're provided with larger geographic zones, seed zones. So they were generally, but in general, all the seed zones were enlarged. So they were fewer and less restrictive than the initial seed zones, and mostly expanded in a north-south direction, but those elevational guidelines were mostly unchanged. This idea of seed zones is also familiar to gardeners, and the USDA plant hardiness zones, or those the sunset guidelines for gardening. And these zones generally were based on minimum winter temperatures of about 5 degrees centigrade. And then with increased interest in restoration, there began to get increased interest in seed zones for native shrubs and grasses and forbs used for restoration in the 90s and in the last decade. And so with that idea in mind, the first seed zones for a grass species were published by Vicky here and working with some folks at the PNW Research Station for Elmus glaucus. So Vicky really gets a lot of the credit for taking these ideas based on forestry and genecology from Bob Campbell and Frank Sorensen and applying them to a grass species. So in summary there, the first seed zones were just basically delineated by the best available knowledge, but not really any genetic studies until they came along and kind of adjusted those seed zones. But now what I'd like to do is talk a little bit about genecology studies and how we use those to delineate seed zones and seed transfer guidelines. So first, the definition of genecology is the study of interspecific genetic variation of plants in relation to source environments. So it seeks a correlation between plant type and habitat type, and consistent correlations are taken to indicate adaptation as determined by natural selection. So presumably, those parents that have the traits that allow them to survive and reproduce produce offspring that have those traits. And when we put all those offspring together, the progeny, into a common environment, we can see those differences. As an example, I used the Douglas fir genecology study, a study I did a while ago. So what we do is we collect seed from many populations and grow those families or populations in a common environment and measure them for many adaptive traits, for example, bud burst in this case. And then we look at that relationship between traits and source environments, and we can come up with a model of that. And then we can use GIS to show that relationship, knowing the environmental variation across the landscape, and map that genetic variation. And so what we find, for example, with Douglas fir is, first of all, that there are population differences for different adaptive traits, or putatively adaptive traits. So for example, Douglas fir differs in cold damage, bud set, biomass, bud burst. Those traits are correlated with source environments. The correlation is most strong, 0.79 for between cold damage and winter minimum temperature, for example. And those traits, those relationships make sense. trees from higher elevations have lower cold damage. And then we also see that different traits show different patterns and scale of adaptation. And what we can do is we can take those different traits and we can use principal component analysis or multivariate statistics to combine them into a few uncorrelated traits. So in this case, vigor, which is essentially related to size, bud set, emergence, some other things, and a uncorrelated second variable, basically it's bud burst and partitioning the height to diameter. And since they're uncorrelated, we can overlay those two and we essentially come up with seed zones. Now this isn't the seed zones that we use for Douglas fir, but the good news is basically pretty close to what we are using. And I'd also take into account that there may be things that we haven't measured, for example, Swiss needle cast. So you may want to restrict some of those transfers a little bit more. But this is one way we come up with seed zones from genecology studies. And so we've done this with Bluebunch wheatgrass, where we've grown them in a common environment, many populations from the Columbia Plateau, Blue Mountains, Great Basin, Snake River Plain. And we grew these in 3 different sites, and we came up with different traits, and we looked at those traits using principal component analysis, and they essentially correspond to 3 traits: size, phenology, heading date, that sort of thing, and leaf width. So we can draw maps of these traits, and since we're using principal component analysis, they're uncorrelated, we can then take those traits and overlap them and come up with seed zones. So these are our seed zones that we've derived for Bluebunch wheatgrass. And what we find, for example, is that small early populations from hot dry sites are smaller, have earlier flowering and narrow leaves, and populations from cooler, more mesic sites are larger, have late flowering and wider leaves. And this is what we might expect based on hypotheses of adaptation to drought and cold. And so we've done this, that's in the bigger sense of we, have done this for several other species. A lot of these have been done by R.C. Johnson at the Agricultural Research Service in Pullman, the Plant Materials Center there. So for example, these are some examples of other species. Mountain brome, these are seed zones derived by R.C. For the Blue Mountains, particularly the National Forest in the Blue Mountains. That's where the samples were mostly taken from. Indian rice grass across the Great Basin and Colorado Plateau, seed zones. Taper tip onion in the Great Basin, Snake River Plain, and Blue Mountains. And one that we've done, prairie June grass in Oregon, Washington, and Idaho. So we do have these seed zones for many species. In the Pacific Northwest, we've now, in the last 10 years, have done 32 genecology studies. And again, when I say we, I'm not talking about me, I'm talking about the larger we. And so 32 genecology studies have been completed or underway for 16 grasses, 8 forbs, and 8 non-timber tree species. And these are at various levels of completion. So one of the issues about genecology studies is they assume that local is best. So we really haven't tested that. But I think that's a pretty fair assumption. There was a reviews by Limu and Fisher and Hereford in 2008 and 2009. And even though there's some issues with the reviews, and even though you try to generalize too large when you're trying to look at a large number of plant species, a large number of animal species, what they found, both of them found, was that the local populations were better than the distance populations 71% of the time. So I think it's a fair assumption that local is best. And where there are exceptions, it's mostly for small populations. And in those cases, genetic drift, which is basically an issue about chance of sampling, randomness, it can limit the efficacy of natural selection. So in general, I would say that the default, more conservative approach is to assume that local is best. The other question is how local is local, and scales of adaptive variation may differ among traits, as we show, and among species. And there's basically two ways that these that we've looked at scales of adaptation. One is called, and I'm not going to delve into these right now, but one is the risk of maladaptation, which is basically a function of the difference between two populations and the variation within populations. And the other is a statistical approach, least significant differences, that's used mostly by Jerry Rayfeld. Now, both of these, though, when you talk about how much of a difference is important, they still pretty much depend on some idea about anecdotal evidence about what degree of a risk of adaptation, for example, is appropriate. And we find, for example, that a risk of adaptation about 30% is what we find within seed zones that we are currently comfortable with. So just as an example of how scales of adaptation differ among species, Jerry Rayfeld looked at the distances needed to detect these genetic differences in the northern Rockies for several species, and what he finds is that some species like Douglas fir and lodgepole pine, you only have to move things about 200 meters in elevation or 18 and 20 frost-free days to find these differences. So they're specialists, whereas other species like western red cedar and western white pine are generalists, and you can basically move them pretty far elevationally or in terms of frost-free days. So what are the advantages and disadvantages of a gene ecological approach? First, they may be done in a short time frame. So generally, we do them in over 1 or 2 years. Because we control the environment a lot, it greatly minimizes environmental variation, lets us see the genetic variation. Since they're small, we can sample many source locations from a wide range of environments and then use those to produce these maps of adaptive traits and climates for easy visualization and manipulation. But it assumes we've measured all the most important adaptive traits, so you may want to consider traits that we haven't measured, for example, disease resistance. It may be difficult to synthesize results of many traits, but again, we use the multivariate approaches to do that. It assumes that local is best. Again, I think that's mostly a valuable assumption, or at least starting point. Also, we're not certain how large a difference in traits is biologically significant, but we have an idea. And it's not a direct test of adaptation. So this is where reciprocal transplant studies come in. Reciprocal transplant study is where populations from a range of source environments are evaluated in the same or similar range of test environments. And the classic example is from Clausen, Keck, and Heisey, where they looked at species like potentilla from 3 different elevations, planted at 3 different elevations. And they, in general, found that at the low elevation population did best, and at the high elevation population did best. So an advantage of reciprocal transplant studies is that we can generate models to predict adaptiveness, survival, growth, productivity, as a function of planting environments. That's called a response function. Source environments, that's the same thing that we've done earlier, genecology functions. The difference between planting and source environments, those are referred to as transfer functions. And look at both the planting environment and source environment together, which has been called a universal response function. So again, an example from Douglas fir, we're doing a reciprocal transplant study. It's about 7 years in the ground now, with 120 families from 60 populations from 12 diverse regions planted back in 9 of those regions. And we had the Genecology study that we did before, so we were able to efficiently design that study and choose populations based on that previous Genecology study. And so as an example of these response functions, this is a response surface for 4-year height. So basically, height growth as a function of the spring temperature at the planting site, the environment that it's growing in, and the frost period at the seed source, the environment in which natural selection occurred. And we can create this response surface. And we can rotate this surface so we can see, for example, look at the effect of planting environment, growing environment. And as you expect, trees grow better in warmer sites and grow more slowly in cold sites. But we can also rotate this the other direction and look, it's more easy to look at specific sites here. So for example, one of the specific sites, Flores, which is a warm site, we see that sources with a frost-free period of about 300 grow best at that site, and that basically shows that things are mostly locally adapted. At Doorstop, which is the cool site with a frost-free period of 190 days, again, about the same seed sources from that 190 days grow best. So sources from similar climates are growing best at each site, evidence for local adaptation. Now we're repeating this, we're doing the same thing with Bluebunch wheatgrass. So we just established this fall reciprocal transplant study with this, using a subset of the populations areas that we did for the Genecology study. We're doing this in 2 transects, one from the hot, dry Columbia Plateau up into the cool, moist Blue Mountains, and the other from the hot, dry areas of the Snake River Plain up into the cooler, more moist areas of the mountains in the Great Basin. And so these, again, come from a diversity of climates or ecotypes and planted back into a diversity of climates and ecotypes. So the advantages and disadvantages of reciprocal transplant studies are that they're a direct test of adaptation, and we can test hypotheses of local adaptation, and we can better model variables of direct interest such as stand productivity. But the disadvantage of reciprocal transplant studies is it takes a long time for results. They're expensive to test over many planting sites and seed sources, and I can tell you from experience this fall that it's also a lot of work. Few samples make it difficult to interpolate between locations to adequately model response or transfer functions or draw maps. That's one of the advantages of the Genecology study. And it doesn't, generally if you don't pay attention to it, which people often don't, it doesn't provide information on the components of adaptation, that is the traits that may be most useful for selection of populations or individuals. So just some generalizations from what we've learned about adaptation of forest trees. And we're starting to learn more about other native plants, but right now I just want to talk a little bit about forest trees. So general forest tree populations are at least moderately locally adapted, although evidence from the few reciprocal transplant studies showed that some populations do show adaptive lag. Species show different patterns and degrees of local adaptation, so if you know something about one species, doesn't necessarily mean you know the patterns about the other species. Most forest tree species show significant variation for timing of bud set, bud flush, cold hardiness, drought resistance, and growth. And those traits would correlate most strongly with minimum temperatures, other variables related to minimum temperatures like frost-free days, and drought indices. So in general, these patterns reflect adaptation of annual growth and dormancy cycles to local temperature and drought regimes. Phenology is important adaptive traits. And in general, provenance tests of forest trees have maybe showed broader adaptation than indicated by Genecology studies. So Genecology studies can be a little bit conservative. But that, on the other hand, provenance tests may show this broader adaptation because they may depend on age and rare climatic events for some of these maladaptations to show up. So to summarize this part, so the experiences with seed zones early on with forestry is that basically we've proceeded with using the best available knowledge. So we've assumed local adaptation, and we've assumed adaptation to climate, predominantly cold and aridity. And then we've used those to delineate seed zones based on climate with considerations for other factors such as disease. So we may not have known everything, but we sat down, basically they sat down and mapped and drew lines on the map just with their best guesses. And then we've used genecology studies to study the patterns and scales of adaptation and variation for specific species. In the case of forestry, then they've revised some of those seed zones and seed movement guidelines. In the case of native plants, we are proceeding right to that. And then we can use reciprocal transplant studies and monitoring in the field to help refine those seed zones and population movement guidelines and help provide some long-term answers. So now we'll move on to Andy for some questions.
There's a question about your data sources for climate data. Can you tell them what your preferred data sets are to extract climate data?
Okay, so there's several. Okay, I'm not sure if you heard that, but so we'll take some questions for clarity right now, and then more questions at the end.
What are the preferred data sets where you extract your climate data?
So the question is, what are some of the preferred data sets from which we extract our climate data? There's several options out there that models that interpolate between weather stations to get climate data for specific locations. The early ones were with Prism. Generally, what I've been using lately, what several of us have been using lately, is something called Climate Western North America, which you can Google that, Climate WNA, and it basically uses Prism, but then adjusts it for elevation to come up with a little finer interpolation between the weather stations. So, okay.
Can you bring that back up?
Yes, I should have done that yesterday. This is bad.
Can you bring that other window up?
Do people see our computer, the problem?
Yes,
Yeah, I can see it. Okay, it's gone.
Okay. All right, we got it. Okay, any other questions for clarification?
There's one other question.
Yeah, I'll get that.
Okay, so Andy will go part 2 on generalized provisional seed zones.
Okay, thanks, Brad. So Brad just explained the process that that's generally been followed to develop the seed zones for specific species. But so the problem is, what do we do about all the other species that are used in restoration that we don't have this information for? We don't have the information on genetic variation. So one thing that the three of us have worked on over the last several years is developing these generalized provisional seed zones for native plants based on climatic data. And so this work was published last year in Ecological Applications, and the maps, the seed zones are all available online, and I'll bring this up at the end to let people know where they can find this. So to build these provisional seed zones, basically these are independent of genetics. As I mentioned, these are intended for species where we don't have the specific genetic data available. We don't know anything about the genetic variation within these species. And these provisional seed zones are really a starting point. With these provisional zones, then it's important to use local knowledge about your area or species-specific knowledge about the particular species that you may be working with in adjusting these seed zones for use. So the data that we used to build these seed zones, as Brad just mentioned, so the data came from PRISM, and the data used 30-year climate normals, which were the period from 1980 to 2010. And it's a continuous data set with 800 meter by 800 meter pixel size that covers the entire continental United States. And the data that we used was minimum winter temperature and aridity, which was calculated as the heat moisture index. And then from this continuous data, we delineated discrete bands for use to actually delineate the seed zones. So for example, here is the continuous data for minimum temperature, where the lightest colors closest to white would be the warmest, and the darkest colors closest to black would be the coldest areas. So this continuous data then was delineated into these 5-degree temperature bands, and the reason that we chose the 5-degree bands was because these match or correspond to the USDA hardiness zones that Brad showed earlier, which are familiar to gardeners and land managers because they've been around for a long time. And then, so we did the same thing with this annual heat moisture index, which is essentially a measure of aridity. And you can see here the darkest red would be the driest areas, and then the green would be the wettest areas. And so then using GIS, we basically did the intersection of the temperature and aridity data, which then delineated these provisional seed zones. And basically what these indicate is that the areas within the zones, which are illustrated by one color, are areas of general climatic similarity. They were within 5 degrees of temperature and within a certain range in aridity. So, but one of the issues is that areas that are similar climatically aren't necessarily going to be similar ecologically. And this is especially evident here in the eastern United States, where you have these large provisional seed zones that go all the way from the Midwest out to the Atlantic coast, basically because the climate there in that area is fairly consistent, which is obviously much different than the western United States, where the heterogeneous mountainous climate really makes the seed zones much smaller. But so obviously the areas in the eastern United States, from the Midwest to the Atlantic coast would be very different ecologically. So then what we recommend is using these the Omernik's level 3 ecoregions on basically laid over top of the provisional seed zones to help distinguish areas that may differ ecologically, even though they are similar climatically. So then if we look at the provisional seed zones with the level 3 ecoregion boundaries drawn over the top, this is how you could then use the combination of these two to help guide seed movement. So for example, maybe in this green area, you may feel comfortable moving material within the green area in here or the green area in this ecoregion, but you would want to be very careful about moving material within this green area between these two ecoregions, because there's some evidence that these areas differ ecologically. Now, the caveat to that is that if you're working in a site that maybe is very close to the ecoregion boundary, or even an area that is within the ecoregion but is very close to the seed zone boundary, this is where your local knowledge and just judgment comes into play, because it may be fine to move things just across an adjacent ecoregion or seed zone boundary, because again these are discrete lines drawn over a continuous surface. So for example, moving material within this ecoregion from the green area to the blue area, even though they're different seed zones, may be preferable to moving material from maybe further north within this green area to the southern part in that green area. So once we developed these seed zone maps, we did some analysis to validate the usefulness of these seed zones. And what we did is we used data from some of the common garden studies. We used data from 2 grasses and 1 forb. And we looked at a suite of traits that are potentially related to adaptation. This included phenology traits, morphology, and reproduction traits. And we compared the level 3 ecoregions, the provisional seed zones within level 3 ecoregions, and the empirical seed zones that were actually delineated from the common garden data. And so this table comes out of the publication, and there's a lot of information on this screen. But really, the point that I'm trying to make here is that when we compare the level 3 ecoregions and the provisional seed zones, so if you look within that yellow box, the level 3 ecoregions, there was for that the allium study, the samples fell into 5 different ecoregions or 9 different provisional seed zones. But the majority of those traits, provisional seed zones accounted for a larger percentage of the variation than level 3 ecoregions. So what this is showing is that the provisional seed zones really help to show where things are adapted to better than just the level 3 ecoregions. And then if you look at the third column, which are the provisional seed zones within the level 3 ecoregions, so this is basically what I was talking about before, where you lay the ecoregion boundaries on top of the provisional seed zones. Those numbers that are in bold there show that those explain the highest percentage of variation between all 4 different classification schemes. And so you can see that the provisional seed zones for several traits do just as good or maybe even better than the empirical seed zones. So we take this as evidence that in the absence of the species-specific data on genetic variation, the provisional seed zones with the level 3 ecoregions really do a pretty good job on helping delineate seed zones or seed movement guidelines for just any species. So now when you look at this map, though, especially in the western United States, it looks very complicated. The intersection of the 2 climatic layers delineated 64 seed zones across the entire continental United States. Now, if you drill down a bit and focus to this, these would be the seed zones just for the Pacific Northwest, for Oregon and Washington. It still looks fairly complicated. There's a lot of colors there, but you begin to see that the majority of the areas really fall into a fairly limited number of seed zones. You know, you've got the pink and the light and the medium green, the light and medium blue, and the purple. So when you start to look at an area that may be comparable to an area where you may be working, it starts to look much less complicated. And so this is a map that someone in the I believe it was in the BLM produced, and this is basically eastern Washington, eastern Oregon, and Idaho here. And so these are the provisional seed zones for sage-grouse areas. And again, now when you focus again, you're drilling down even further to a specific area. I think there's 18 or so seed zones that are indicated, but again, you start to see that the majority of the area really falls into a much more limited number of seed zones, which means that this is going to be much more manageable as far as seed procurement and seed production. And then if you focus even further, drill down, this is the Great Basin, Northern Great Basin, and Snake River Plain Level 3 ecoregions. You see again where the majority of the area falls into a fairly limited number of zones. Different colors in this map indicate different temperature bands, and you can see between the reds, the greens, and the blues, that covers the majority of the area. The oranges and yellows, which would be at the low and the high end, really are a very, very small portion of the total area. And to further simplify it, there's certain areas, say higher elevation or very dry areas, where it's not likely that restoration work is going to be done, or the species that you're working with wouldn't be found. And on this map, what this shows really well is if you look at the Snake River Plain ecoregion, you go from the warmer area near Boise in this red, which would be the 20 to 25 degree band, and as you start going upstream, you get into the green, which should be the 15 to 20 degree, then into the blue, which is the 10 to 15, and into the yellow, which would be the 5 to 10. So you can see how it tracks, the seed zones track the temperature within that ecoregion. So now all of the seed zones are all available online on this website. It's the Seed Zone Mapper. And this is on the WETAC, the Western Wildland Environmental Threat Assessment Center website. And there's this Google Earth-based geo-browser where you can turn on different layers. There's the seed zones for all species that's shown here. There's also the seed zones that have been adapted or modified for the Great Basin. And then on these other tabs, there's several of the empirical seed zone layers. You can turn these layers on and off. You can zoom in and zoom out to your particular area of interest. In the Tools tab, here you can actually input a specific latitude and longitude to zoom directly to a particular spot, if that's your restoration or your project location, or maybe where the seed was collected from. So you can work with these layers here in this geo-browser. But you can also, on the main page, there's also, you can download the GIS shapefiles for the provisional seed zones across the entire United States, or the Great Basin, the ones that have been modified, and then several of the species-specific, the empirical seed zones that Brad had mentioned and that he showed some of the maps earlier. These can be downloaded here, as well as PDF maps and the publications that were put out for each of these species. And so up here on the top of your screen is the URL for that seed zone mapper. So I'll end with just a few caveats about the provisional seed zones. First of all, obviously microsites need to be appropriate for the species. So just because you're within the same seed zone and even the same seed zone within an ecoregion, you're going to use riparian species on riparian sites and not on dry upland sites, and vice versa. Second thing is as Brad mentioned, the seed zones differ by species. There's generalist species and specialist species. So local knowledge is important, both the knowledge of your species and of your geographic area. And then, of course, research is still needed to construct the seed movement guidelines based on the actual genetic data. You know, these provisional seed zones are a starting point. The idea is that they're better than no information at all, but these obviously don't supersede these empirical seed zones that have been developed based on the species-specific data on genetic variation. So now I'll hand it over to Vicky for part 3.
Are there any questions?
So the questions for Andy mostly had to do with where to find those seed zones, and I think Andy answered most of those questions for clarification with respect to the seed mapper tool. So, Vicky.
Hello, everyone. In this third and final piece of the Seed Zone webinar, we're going to shift gears a bit and now focus on some of the more practical applications and the benefits of using seed zones in our operational plant material programs and revegetation activities with special emphasis on the Pacific Northwest region of the Forest Service. So just for those that may not be familiar with our region, the National Forest and our one national grassland occupy roughly 25 million acres throughout Oregon and Washington, occupy just a really diverse range of ecosystems and vegetative types from east to west, north to south. It's an area where disturbances are very common, especially in the more fire-prone areas of our region in southern Oregon, and then on the east side of the Cascade Mountains in eastern Oregon and Washington. Throughout the region, there's just a major emphasis on restoration for a variety of purposes, which I'll get into a little bit later. Because of our land base and other factors, the species and the seed sources that we want to use in our revegetation activities, I would say for the most part are not commercially available on the market, in the marketplace. And so about 15 or so years ago, the region implemented a native plant material program to provide for our internal restoration needs, with the idea being to have a seed bank in place with the appropriate species and the appropriate seed sources for when and where we needed it throughout the region. So it's really very similar in concept to the way we operate our conifer seed program for reforestation. And just like with conifers, seed zones are just foundational to our native plant material development efforts and are integrated into nearly every aspect of our program, as shown here in this cycle, seed production and use cycle, from seed need planning through wildland seed collection and production, our testing and storage operations, and then to deployment, seedings and plantings. So what I'd like to do now is just kind of go through each step of this cycle and talk a little bit about how we're utilizing or integrating seed zones into these various activities. I'll start with seed need planning, because really I think this is where it all starts and is a very important piece of the puzzle, and focus on 3 levels of planning: a broad scale or strategic planning, more project level type planning, and then the wild card, the unplanned disturbances. So to start out with the broad scale type strategy sort of documents that are continually coming online, I feel that seed zones can serve as a really useful framework for us dealing with both our internal and external partners. Oftentimes these strategies cross multiple land ownerships. So it really gives us a framework for working with those folks to plan and coordinate plant material needs that are associated with these various strategies, initiatives, and prioritization analyses. A good example for us here in the Northwest is our Pacific Northwest Aquatic Restoration Strategy, which was developed several years ago. It identifies high priority watersheds across the region, which will be the focus for years to come for aquatic restoration work, things like putting roads to bed, culvert replacement, and so on. And so these priority watersheds are a good place for us to focus some of our plant material collection development efforts, because we know that there's going to be a tremendous amount of activity in those various watersheds for some time to come. Some other good examples, I think, coming online would be the new USDA pollinator strategy. Here again, I think seed zones can serve as a useful framework for us to work with other landowners and partners in developing plant materials for pollinator habitat improvement. Andy mentioned, and Brad did too, the efforts going on with sage-grouse recovery. And there again, I think seed zones can serve as a really useful framework for developing plant materials for sage-grouse habitat improvement. Stepping down to the project level, as I mentioned, in this region, restoration is king for a variety of reasons. I mentioned aquatic restoration, so there's a lot of road decommissioning going on, culvert removal, dam removal, also wildlife habitat improvement projects, energy projects, invasive plant management. And so the idea here is that the revegetation in these projects and plant materials is not an afterthought. It's something that's planned in advance so that the appropriate species and plant materials are in place and available for these projects. And so each district or forest the idea is that they have a plant material coordinator that's working with their engineers or fish and wildlife biologists, invasive plant managers, and so forth, to understand what their needs are going to be, where they're doing projects, and work with them to secure funding, and then collectively go into a plant collection and propagation effort to make those materials available for their projects in out years. This is an example of one forest kind of template that they're using to identify those projects. So on the left, they're laying out by fiscal year the various types of projects. They just cover a whole array from timber sales to aquatics projects and so forth, laid out across the multiple fiscal years for as far as they can foresee into the future. And then across the top would be the various kind of the major workhorse species that would be utilized in those different types of projects. And you can see where there's species-specific, empirical seed zones that are available. Those are identified for the different species. And then otherwise they're nested in within the provisional seed zones. And then I've just populated this with some dummy numbers, but basically they're just trying to identify for each of the projects the amount of seed that they think would be needed for that particular project and the different species that would be utilized. Then those can be summed up here at the bottom across the different species and seed zones, and then compared to the seed on inventory. And if there's a shortage, then collection and propagation plans can be implemented. So I mentioned earlier that wildfires are kind of the wild card. We do a pretty good job at our known and planned projects, but it's wildfire which drives so much of our restoration needs that we're really trying to get a better grip on. Especially in our conifer seed program, we're kind of pioneering some of these efforts, but the same sort of approach could be used for other types of plant materials. So this map just shows, actually this is in Central Oregon, and we're overlaying historical fire occurrences, superimposing that with our seed zones to try to get an understanding of where fires tend to occur on the landscape. They are not random. They do tend to occur in certain geographic areas, and so these are historical glimpses can give us a sense for where we need to be focusing some of our seed collection propagation efforts for wildfire use. Another and more probably sophisticated approach would be to take advantage of some of the wildfire risk modeling that's going on in this region and elsewhere. On the left, you see for Oregon and Washington a wildfire risk map, which is then on the right superimposed with our conifer seed zones to get some, and then some mathematical formulations are going on behind the scene to come up with an expected number of acres burned annually by seed zone that could then be converted into a 10-year seed plan for conifers for each of those zones. Okay, moving on to seed collection, propagation, and storage. Here again, seed zones are just integrated into every aspect of this work, starting with seed collection. This work, for the most part, or my preference is to do this in-house, but we do have a regional IDIQ restoration services contract in place that includes a bid item for seed collection. And what you're looking at here is just a portion excerpt from a task order for seed collection for multiple elevational bands of 2 species, blue wild rye and Elmas, or blue wild rye and mountain brome. And again, it's all organized by seed zone. So the contract would contain, besides stipulating the amount of seed to be collected for each of those species in each of those seed zones, the contract would stipulate the number of populations to be sampled, the minimum distance between those populations, the number of individuals per population, and how that plant material should be bulked. Those are Kayla Harriman in a subsequent webinar, I think in February. We'll be talking in more detail about some of those best management practices and methodologies for seed collection, but I just wanted to, I guess, point out using this map that regardless of the details of the collection, that seed origin and that seed zone coding would be labeled on every bag that comes out of the forest after collection. In our region, that material would all go to our Bend seed extractory in Bend, Oregon. So they extract, clean, and store the seed for our region as well as other Forest Service regions in the western U.S. and a number of other federal clients such as the BLM, Fish and Wildlife Service, Park Service, Federal Highways, BIA, and tribal entities. So they have a wide array of clientele, and they are available for other federal business. They have tremendous experience processing over 2,700 native plant species to date. That's the most of any extractory that I know of anywhere. They, again, like I mentioned, they maintain our inventory and provide some amount of granary storage and then freezer storage for all our wildland collected material. And again, all that material is labeled and stored using that initial seed source identification code that's assigned to that material at the time of collecting. From here, I might go to one of our federal nurseries. We're very fortunate to kind of sit in the center of 3 outstanding federal nurseries, J. Herbert Stone Nursery in Medford, Oregon, Lucky Peak in Boise, and then Coeur d'Alene in Idaho. They all do native plant material, seed production, and bare root and containerized plant production. We worked extensively over the years to cultivate our native seed producers. In the western U.S. Pacific Northwest, we have a regional IDIQ seed production contract in place that's heavily utilized by forests in the region. Is also available to the BLM and Forest Service units in Idaho. So here I would just say that like our seed collection, that grow-out work, it's just all organized and labeled and done very carefully using seed zones as kind of the primary driver for how that material is propagated and produced by both our federal nurseries and the private seed producers. Doug Kendig with the BLM will be talking in more detail in a subsequent seminar specifically on seed production IDIQ contracts, so I won't belabor this, but just wanted to say that for us this is just a very powerful vehicle for us to produce high quality plant material from experienced seed producers with strong controls and reduced costs, and again it's all driven by seed zones. When it comes to deploying that material, as I said earlier, hopefully the project is planned and there's plant materials available and waiting for use when the project's ready to go. But we know that all the time there's projects coming along that weren't in the works, that weren't planned at the time plant material was developed. But hopefully there's inventory on stock, and what people do would be go into GIS and they can overlay their project with seed zones, in this case provisional seed zones. And I'm just in this example just showing you this is actually a federal highway project going through I-90 corridor in eastern Washington, just showing 3 different project areas within that larger project site, and fortunately all of 3 of those pieces lie within one seed zone, and plant material would more than likely be available for that project at this scale and within that one zone. This is an example of a more complex project. This is a proposed pipeline route that goes through western, all the way from the Willamette Valley, across the Cascades and into eastern Oregon. Just an example of crossing many different provisional seed zones. This would be a project where, again, the district people would lay out the map line of where that project was going, look at their seed inventory, and then work with the energy company to be reimbursed for plant materials that might be utilized in this site that we already have in our inventory, or to arrange for like cost share agreements where we would be reimbursed to go out and do collection and propagation work specifically for projects such as this. So seed zones also, I think, in having knowledge of plant material origin can play a huge role in helping us understanding the effectiveness of our revegetation work and the influence that seed sources might have on the initial survival, the growth rate, or reproduction of that plant material years out. And this is, I think, an area where we have a lot of work to do. We don't quite yet have the systems in place to really be doing this yet operationally, but it's something that we're striving to do, and I think it's just going to be all the more important with climate change to be able to understand better how these different seed sources are performing out there in our different types of projects and settings. And that kind of leads me into the last few slides here, which I just wanted to talk a bit about how seed zones can be a tool, I believe, for helping us in responding and adapting to climate change. For starters, I think seed zones can be a very useful framework for helping us communicate to our resource specialists about current climate within our seed zones, as well as where we believe changes are going to happen in the future and where we need to focus our efforts, perhaps most importantly. On the left is an example in California where they're mapping out just in the red and the yellow kind of their hottest, driest zones, which maybe would be areas where they would do more focused monitoring to understand if they're starting to have problems or when they're starting to have problems. Might be kind of some priority areas where they would look at to perhaps start implementing mixes of different species perhaps, or different seed sources that have been used in the past. On the right, we're showing for our conifer seed zones, which are for us, because they're long-lived species, of greatest significance to us in terms of looking ahead at climate change. That map is an example where we're projecting mid-century summer temperature increases in our Pacific Northwest conifer seed zones, again, to get an idea of just where changes are predicted to be the greatest mid-century. As you can see, probably no big surprise at the scale this map is displaying anyway, that in the east side of the Cascades, especially in the southeast Oregon area, that's where the problems are going to happen soonest and be most pronounced in the near future. We can also use our seed zone framework and look within seed zones to perhaps give us some ideas about how we might modify our seed movement guidelines for the future. This is an example of some work done by Francis Kilkenny now at the Rocky Mountain Research Station, and he's looking at mean annual temperature ranges across 1,000-foot elevational bands here at the bottom within, this is just a single Douglas fir seed zone, just as an example. And so you can see, well, within each of these boxes, the dividing line between the blue and the orange represents the median temperature in that seed zone, and then 50% of the seed zone would be encompassed by the temperature range barred by the blue and the orange band. And that's projected or shown for current climate, which is defined as that 1961-1990 time frame, and then projected out for that same 1,000-foot elevational band to the year 2050, mid-century. And so what this is showing is that within each of these 1,000-foot elevational bands, by 2050, we're projecting some, 1/2 to 2 degree shifts upwards in temperature within each of these bands. And how we're using this right now, and this analysis needs to be completed for all of our zones, but this can be a tool to work with our silviculturists, for example, to show them that perhaps if you're looking at plant material that's originating from a site, say in this under 1,000-foot elevational band, that by the time 2050 comes along, it might not be the best seed source suited to that site. Yet if it were to be moved up in elevation, say 1,000 feet by 2050, that would be a much better temperature band fit for that plant material. And that just extends all the way across every elevational band. So the idea being that we might start to be looking at utilizing plant material or mixing plant material with local sources, moving at, say, 1,000 feet up in our reforestation plantings. Francis Kilkenny, this is, again, his work is with Bluebunch Wheatgrass, and he, again, will be giving a subsequent seminar, I believe, in March, strictly on climate change and using genecology research in this context. So he will be speaking more in more detail on this. But I just wanted to point out, again, this is kind of becoming the standard way now of taking current climate and genecology seed zone products, but manipulating them and forecasting them out to the future to look at how things are predicted to change, and so that we can be thinking about that and using that as we go forward. This example is a predicted change in Bluebunch wheatgrass heading date in the new seed zones that have been developed for that species in the Great Basin. So as just an ending slide here, just want to reiterate some of the benefits as I see them of seed zones. First of all, they help us meet our policy objectives, as Brad started out with some of our policy statements regarding use of plant materials. It also allows us to be more successful, I believe, because in our restoration success, because we're using adapted plant materials now and into the future. It helps us increase efficiency. The map on the right is showing our Bluebunch or our blue wild rye seed zones, the 4 of them for northeastern Oregon, superimposed on the old framework we used, which was our fifth bill watersheds. There are hundreds of those. And so substantial reduction in the number of management units for which we're having to plan and develop plant materials for. Those zones tend to cross administrative boundaries within the Forest Service as well as other land ownership boundaries. So it really puts us in a good position, a much better position than the watersheds for sure, to be able to share and exchange seed among those administrative units and other landowners. That economy of scale, where you're producing larger seed for a given zone, certainly helps reduce costs, and perhaps also would entice additional growers to want to produce this kind of plant materials, which will help reduce costs in the long run, I believe. And then a final point, I do feel that seed zones are helping us to increase both the supply as well as the use of seed and our overall restoration capacity in the agency. And with that, I am done.
Okay, thank you so much. So now we'll open the floor for questions. I'll begin fielding questions that have already come in. All right, so I think this one was answered already, but just to be sure, Ian Grinter asks, is there a source for the completed seed zones?
Yeah, so the best source is that Seed Zone Mapper website that I showed the screenshot of, and on one of the slides I did give the URL for that. That's publicly accessible. It's not behind a firewall or anything like that. So that should be available to anyone on the internet.
Okay, great. Thank you. And then I believe that question also addressed the question, is the map online? It has the ability to zoom?
Yes, that's correct. Okay. You can zoom in and out at pretty much any scale. It's Google Earth based, so you can zoom, zoom and navigate the same way as you would in Google Earth.
Great, excellent, thanks. Okay, there are some questions from Mark Skinner directed to Brad, and the question is: you have distinct seed zones based on elevation and climate, so isolated mountaintops are in the same seed zone. These isolates are more closely related to the plants just downslope than they are to each other, correct? Therefore, if you use the seed zones, you are technically not using the most local material, you're using the most adapted material. This seems to violate the idea that local is better, although it must be a matter of scale. And he asks for comments, but hold on a minute because there's a couple follow-ups. In other words, when we say genetically appropriate, we mean better adapted, not more closely related. Therefore, does it mean that when we use seed zones, in some cases we are disrupting the local genetics of nearby populations, assuming gene flow? Done.
Okay, so good question. Genetically, so yes, we do mean genetically appropriate, but we also do mean that they are probably more closely related with respect to those genes that determine adaptation. So, what you're referring to is isolation by distance. Geographically close, they should be more closely related. But that's not necessarily true if natural selection is strong enough to cause divergence in those adaptive traits so that you see divergence between things that are geographically close, but they look different in a common garden study. So, I mean, natural selection must be strong enough to cause that. So they may be, based on isolation by distance, more closely related in general, but with respect to those traits that are important for adaptation, I'd say no. That there must be divergence in those traits, otherwise we wouldn't see differences in common garden studies. Does that make sense?
I think so. So Mark, type in another question if it didn't make sense and we'll get back to it. Okay, thank you. Next question is from Adrian Pilmanis, who asks, discuss cost per pound of grass forb seed versus commercial, considering all planting, labor, and infrastructure costs. Compare with the environmental costs, direct, indirect, cumulative impacts if quality seed is not used in restoration projects.
Well, I will address the first part on cost. First of all, some of the species that we want to use in our restoration plantings, particularly forbs, they're not available on the commercial market in any format. I mean, you can't buy them. So we have to produce them if we want them. Some of the more common workhorse species, Bluebunch wheatgrass, mountain brome, Elmus glaucus, those are the costs for those that our local material that we're producing we've seen those really come down over the years as growers gain experience and the demand increases. And I'd say those are pretty darn competitive if commercial sources exist for those plant materials. But again, in big fire years where some agencies might be going out for big seed buys in big fire years, we've seen where our local material that we're propagating through private growers or even at our in-house facilities, the cost of that material can be actually less than what we're seeing prices on the commercial market. So, it really can be all over the board, I guess. In terms of environmental costs, well, I'd say they're pretty huge. Some of the traditional cultivars, the single source cultivars that lack genetic diversity that have been used for decades across much of the western U.S., such as Anatole, Bluebunch, Wheatgrass. I mean, think of the impacts of the diversity at the species level that's happened to that species that we really aren't even able to really track at all, but you know that it is occurring. So, so that would be my answer.
Okay, thank you so much. All right, that looks like the last content question. I do have a question about whether or not these webinars will be recorded and so that they can be viewed later. And yes, all the webinars will be recorded. I'll get to that in a second. Let me make sure. Yeah, that is the last question. Wonderful. We would greatly appreciate it if you would take our 3-question survey of this webinar that will appear after the webinar has ended. And I want to let you know that a recording of this webinar will be sent to you automatically through the GoToWebinar system tomorrow, and then I will also post it on our Great Basin Fire Science Exchange website along with a PDF of the PowerPoint presentation within the next couple of weeks. I'll try to be quick about it. And if you have more questions regarding this webinar or have requests for future webinars, please email or call me at any time. Let's see. Great. Thank you for the positive comments, and thank you all for your participation today. And thank you, Brad, Andy, and Vicky, for your presentations.
Yeah.
Thank you.
Thank you. Our pleasure.
Yeah.
All right. Have a great day.